All right, welcome everyone. My name is Jeshuin, and today I'll be talking about some of the hackathons we have going on, specifically the BioML Challenge 2024, and submission deadlines for that is September 30th. That might be a little bit soon for some of you guys. Team signups for this were due back in like August 23rd, so um, I do have a team set up, but for future reference, I will be posting these a little bit more frequently. But yeah, so let me just catch you guys up to speed. What exactly is this challenge? What are we doing? So basically, we're designing a binder to target this one guy. And who is this guy, you may ask? This guy is CD20. So CD20 is a protein that's normally found and expressed on B cells, and it's important because it helps regulate how B cells grow and respond to threats when the body encounters things like bacteria or viruses. However, in certain cancers like B cell lymphoma, these CD20 things can get overrepresented and, they'll just be all over the cell. So since the CD20 protein is over expressed and it's all over the B cell, it's actually a good target that we can use to throw in our binders with this thing called CAR T cell therapy. So one thing we need to keep in mind is since these are on normal functional cells, if we have the binder design too strong to that, then what would happen is it could target healthy and normal cells, which we don't want. So we kind of want this like Goldilocks type situation here where our binder isn't too strong, but it isn't too weak either. So that's something we have to keep in mind. And how exactly are we going to do this, you may ask? Well, great question, because we're going to be using a bunch of AI tools to help speed up the process. So normally this process of designing a binder, designing a construct, or whatever you may call it, this is a very expensive and arduous process. It takes a while, it takes some money. So because we have some AI tools like RF diffusion, AlphaFold, it can help speed things up. And these are all computational tools that you can run. I will be posting everything on the GitHub. I will be posting more videos so you guys can understand everything in more depth. If you guys have any more questions, feel free to talk in the Discord because I will be answering them to the best of my ability. If not, other people will. It's kind of like a big learning thing. So yeah. Okay, and for the next challenge slash project hackathon thing, we have something called Marco Bolo, which is focused on improving our understanding of the health of our oceans, our lakes, our coastlines, our water ecosystems, right? It's funded by Horizon Europe, and it aims to find better ways to observe marine biodiversity in our ecosystems. So what exactly is this challenge and what are we doing? So one of its main goals is to use eDNA or environmental DNA data to help us validate this fact of biodiversity. So when animals live in water, any other organism, we shed skin, we have scales, we release waste into the environment. And analyzing that from a water sample could be quite difficult. And there are a few tools that I've looked up. I'll leave them up right here. QIM2 and DADA2 are pretty popular when interpreting eDNA data. There are some other tools out there that I hope we can look into, and the deadline for this project for team formation is the 13th of September. So everyone that wants to participate in this, please go ahead and check out the Discord, sign up. And there is a info meeting this Monday, actually. I believe it's 9 a.m., going over any questions that people may have for the project. And yeah, so those are the two projects. If you would like to sign up, please go ahead, look at the Discord, look at our GitHub. And yeah, I hope to see you at some of these events. Sign up for everything. Check out our Discord.